Hello, everyone, and thank you for joining us today for SIO's webinar with Dr. J. William LaValle as he presents on the practical understanding of the hallmarks of cancer in molecular integrative oncology. Dr. LaValle provides evidence-based, molecular network-targeted, molecular integrative oncology, MIO, treatment plan recommendations for anti-cancer natural products and repurposed pharmaceuticals in addition to, not instead of, conventional oncology chemotherapy, targeted bio and/or radiotherapy. He has developed advanced data mining tools to access the molecular biology of cancer, peer-reviewed published literature, and genomics, transcriptomics, proteomics databases to search, curate, develop, and update rigorous evidence-based molecular integrative oncology treatment interventions utilizing natural products and repurposed pharmaceuticals. Dr. LaValle has been a member of the Society for Integrative Oncology since 2003 when it began, and currently serves since 2017 on the SIO Communications Committee. Dr. LaValle is an advisory board member of gettingtoknowcancer.org and its affiliated clinical research organization, broadspec.org, and actively supports the mission for evidence-based broad-spectrum approaches to cancer prevention and treatment. Dr. LaValle can be reached at his website at www.lavallemdprotocols.com, which will be placed in the chat. So no worries about remembering that URL. But Dr. LaValle, we are so happy to have you with us today and excited for your presentation.
Thank you, Morgan. Appreciate it. Good afternoon to everyone. Thank you for coming. Welcome to the Practical Understanding of the Hallmarks of Cancer and Molecular Integrative Oncology. This lecture is a fast-paced storyline. It's designed to be fast-paced. So it helps to watch it like a documentary video, and you can watch it again on YouTube if there's any additional information you want to review. And you follow the step-by-step sequencing of this information. It's designed and intended to give you a solid foundation, practical understanding of the hallmarks of cancer. Most of us didn't get a training in the hallmarks of cancer or a deep or broad understanding of the hallmarks of cancer. And then also to touch on and give an overview of the molecular integrative oncology as they relate to the hallmarks of cancer. So that's what the goals of this lecture are. And the emphasis is on molecular networks in cancer cells and in tumor microenvironment cells, and how molecular integrative oncology can be used as a rational, evidence-based supportive treatment for patients with cancer. The hallmarks of cancer, you'll see in the literature this graphic again and again and again. It comes from these two landmark papers, one that was in 2000 and another 11 years later in 2011 from Hanahan and Weinberg, and you'll see it again and again. The hallmarks of cancer are the common attributes, the common features, characteristics, behaviors, phenotypic expression that exist in all cancer types. And so it's important to recognize it covers the broad range of cancer and that they're produced by the molecular activities in the molecular networks in cancer cells and tumor microenvironment cells. And when you look at this graphic describing it, it can be a little challenging to understand, and recognize that although Hanahan and Weinberg have put it into this sequence with an implying that there's a sequential progression, they're essentially isolated particular characteristics that are being described. And what if we can rearrange them into a sequence that actually makes it easier to understand the rationale, the logic of the biological progression? So that's what's being done here. And I did that in understanding the molecular integrative oncology. So there's all the same hallmark descriptions and put into a table format, and then rearranged in a rank order that makes easier for explanation and understanding. So let's start with what we know and then compare it to the hallmarks of cancer. And looking at healthy cells and organs and those same activities in the cancer. First, healthy cells have stable DNA. When you look at DNA in the double helix, the DNA, that the strand of DNA is wrapped around these proteins that look like spools, and they're called histones. And then the histones themselves are gathered and connected into these circular rolls, which are also then coiled on themselves. And even those coils then are put into or defined into these long rolls. And it's only during actual division, replication, mitosis, that we see the chromosome in this familiar format, the chromatid. And the basic understanding of molecular biology is that DNA is transcribed into RNA, RNA is translated into protein. Think of it as the blueprint. The DNA is the blueprint, that software, the instructions, that is then transcribed into templates, and then those templates are then translated into building blocks, the protein. So DNA makes RNA, makes protein. And the three big omics studies, genomics, transcriptomics for the RNA, and proteomics for the protein. And think of it this, is that DNA is this exquisitely orchestrated, responsive, with ongoing waves and bursts of DNA expression. It's not just one at a time. There's an entire symphony of DNA expression occurring all the time with its downstream RNA transcription and protein production. It's concurrent and it's dynamic, it's precisely controlled, and it can occur very rapidly in milliseconds up to seconds. And there are hundreds, even thousands of events going on in every cell all the time. DNA makes RNA, makes protein. And that the metabolism in healthy cells are bringing in the nutrients and managing the nutrients to make various activities within the cancers, within the healthy cells. And that there are growing signals, growing molecular signaling activities. And then when the cells, which are now in their appropriate location in that organ, you can think of it as a geographical location, that when they reach the appropriate size and shape, and they then turn off those growing signals. And those growing signals are then sensitive to being decreased or diminished or turned off. So you're growing and stop growing. And that's normal and well characterized within healthy cells and organs. What's more important or probably less well taught and well understood is the concept of molecular signaling. So this animation, looking very crudely at a molecule that's binding to an external receptor, causes that receptor to change shape, and that the signal then transduces inside to the cell and is now actively adjacent to another molecule, signaling activation in that molecule, which then downstream signals to another, which can activate another or even a complex of molecules, and in this case, demonstrating that the molecule goes into the nucleus and initiating transcription. It's important to describe and understand that molecular signaling is the transfer of information or actual molecular mass. So think of it this way. You have a molecule, and the molecules have various shapes. Sometimes the shapes are very rigid, other times they're quite flexible. And that when you get right to the edge of the molecule, the electromagnetic signature, so the characteristics of the electromagnetics of the atoms within the molecules cause it to have an electromagnetic configuration in that shape, which has influence in the nearby environment. When then that molecule gets close to another molecule, it can then affect the molecular in electromagnetic shape and size of that molecule. Now that molecule can then, under the influence of the first, without even touching it, just getting close enough, can cause it to change shape, can cause it to break apart, can cause it to bind to the first one. There's many different options that can occur, and then this molecule can then downstream signal another molecule, which can then downstream signal another one. So there can be a sequence of molecular signaling from one to the next, to the next, to the next, and that's called a molecular signaling pathway. And sometimes it's very rapid and automated. It starts here and just step down to here very rapidly and completes that molecular cascade. Other times the molecule can get and signal, and now this one, depending on the conditions around it, depending on what other molecules are nearby, depending on the other environmental conditions, may signal this one, may signal this one, may signal that one, may signal that one. So it can go in multiple directions, if you will. And so you can think of that as sometimes the molecular options, it's as if this one is at a molecular intersection. It can go left, right, up, down, or a molecular traffic circle with many different options depending on the circumstances. There are hundreds of molecular pathways in every cell, and the pathways are not separated. The pathways are by definition overlapping and intersecting. And so it's helpful to think and useful to think of this as molecular networks, molecular traffic signaling networks within the cells. And we'll move forward to the next feature, the next characteristic. Normal healthy cells over time can accumulate damage. They can degenerate, they can accumulate products, waste products that are not completely or appropriately digested or removed. And so they can become inefficient and degenerate. And that then there are signaling activities, feedback, that the cell then automatically dies. It's automatic programmed cell death or automatic recycling, so that the constituent components, parts of them, can be reutilized, that the molecules can be reutilized. And that automatic programmed cell death, apoptosis, is well understood. And that the cells in healthy organs, they don't replicate unless necessary. They're have a limited number of replications, and it's a healthy, limited replication. And that there's a healthy blood supply within the organs. If they're, for instance, building more muscle mass, then you need more blood vessels to provide nutrients and oxygen for the muscle mass. Then the cells can induce the growth of those blood vessels. And that the immune system maintains a healthy balance, modulating between good inflammation and good anti-inflammation, maintaining a good immune equilibration. And that when cells do degenerate and become such that they're presenting molecules on their cell surface that identify them as damaged, as deteriorated, or even as malignant, then the immune system, healthy immune system, can find and eliminate those cells, so that those cells are sensitive to immune destruction. And that healthy cells don't invade nearby organs. So they respond to the feedback. For instance, the stomach doesn't grow into the pancreas, or the lungs don't grow into the ribs. What happens is that when a cell in a margin of an organ hits that margin or against another organ, that the feedback is that it turns off the growing, and that there's transition cells from one organ to another, but the cells don't grow into other organs, and they don't release from the organ to take up a location in other locations in other organs and begin growing in other organs. And importantly, the healthy cells have the ability to adapt and adjust to adverse circumstance, adverse danger. So infection, poison, trauma, extremes in environmental conditions. And that healthy adaptation we call resilient, robust well-being, resilient, robust health. Now let's look at each of these categories with the hallmarks of cancer, and looking at the description in user-friendly terms: DNA mutation and instability, pro-cancer metabolism, the ongoing pro-growth molecular signaling, and the lack of sensitivity to turning off the growing. I'm going to explain all of these further in a sequence that actually gives a logic or a rationale to how this occurs. So instead of the growing turning off, the growing just continues. And they're resisting cell death and limitless replication, growing its own blood supply and producing pro-cancer inflammatory molecules and able to avoid immune destruction, and that the cancer cells can invade nearby adjacent organs, as well as releasing and take up and grow in distant locations. And importantly, and unfortunately, the same ability to adapt and adjust to adverse conditions is not eliminated. So cancer cells can adapt to adverse stress, including the treatment, including toxins, so chemotherapy, and develop resistance to adverse stress. And importantly, the cancer cells, because the DNA is altered, the DNA is mutated and unstable. The unstable part is very important, that the downstream progeny of the populations of cells that are produced, they're not 100% identical to the upstream parent cells. And so you get these populations, subclonal populations, so groups of cells downstream that are different, sometimes slightly different, sometimes significantly different than their upstream parent cells, and that's called tumor heterogeneity. That has profound implications with regard to treatment, because some treatments can target the vulnerabilities in several populations where there may be other populations that remain resistant to that. So now let's compare it to what Hanahan and Weinberg said. The DNA mutation and instability is genome instability and mutation. Pro-cancer metabolism, deregulating cellular energetics or metabolic reprogramming. The ongoing pro-growth molecular signaling is called sustained proliferative signaling. Self-sufficiency in growth signaling. So the signaling is turned on autonomously by itself. And the evading growth suppressors. So the lack of sensitivity to turning off the growing signals is evading growth suppressors. Resisting cell death, so they don't automatically and easily undergo automatic programmed cell death and cell recycling, and has this limitless replication called enabling replicative immortality. Can make its own blood supply, inducing angiogenesis, and has tumor promoting inflammation. The cancer cells can produce tumor promoting inflammation as well as induce it in other cells, avoiding immune destruction. We'll show that more in a moment, and then activating invasion and metastasis. And so I find that the list here is easier to discuss with patients and their families, or with other physicians, and then referring it to the descriptions that come from the literature. So here's the association, and notice that this sequence of events is not exactly the same sequences implied here, but you can think of these in the actual literature as independent of one another. Here, we're going to put them together into a sequence that actually makes it more understandable. Healthy cells subject to danger when they become cancer cells, some adverse circumstances, one or more circumstances, induce that transformation. So danger, infection, poison, radiation, chronic inflammation, even inherited characteristics in the DNA that are uncovered or finally expressed, that the mutation in the DNA and the instability of the DNA is important to understand. Again, the DNA is transcribed into RNA, translated into protein, and that gets disrupted and becomes dysfunctional. So this very well organized and highly orchestrated activity, cascade of activity, thousands of events can become very dysfunctional. And the result is you get aberrant gene expression and dysfunctional or aberrant transcription RNA activity and dysfunctional protein activity. That can be aberrant proteins or too much protein, excessive proteins, or too little proteins. The result of which the normal, well-orchestrated gene expression waves and cascades can now become very disrupted, very dysfunctional. And that's a big, important understanding. Then the pro-cancer metabolism. And many of us in integrative medicine have heard that and understand that cancer is a metabolic disease. There's a lot of emphasis about that in the integrative medicine community, and that's absolutely true. Cancer is a metabolic disease, and it's also important to recognize that it's also a disease of the other 9 hallmarks, these other characteristics. And while we put a lot of emphasis on this, that it's important to expand and extend that emphasis to include all of these hallmarks. And when these same kind of dangers, these same kind of adverse circumstances can cause damage to mitochondria and alteration to the mitochondria, that can result in the increased accumulation of oxidation products, reactive oxygen species, ROS, that those can cause mutation, can cause instability in the DNA, as well as the DNA downstream, the unstable DNA activity, as I described earlier, can interrupt and cause adverse activity in the metabolism, and that this can be a vicious cycle, the result of which, whether it starts in the mitochondria or starts in the DNA, the result of which is there's a transformation from a healthy cell to a malignant cell. And that now these 2 hallmark features, the growing signaling now is turned on, and the sensitivity to turning off that growing signaling is eliminated. And so the signaling just continues, the grow continues. And the cancer cells can develop resistance just like the healthy cells, which is described earlier, that ability to adapt to survive, to compensate. It's called compensatory signaling. And the result is the cells are able to persist. They're able to resist that automatic programmed cell death, the next hallmark feature, and accumulate by the getting large enough, triggering replication, and that process continues over and over and over. So you get the limitless replication, and the result of which is you get a tumor, a lump of cells, a malignant tumor. And remember, because the DNA is mutated and unstable, there are populations of cells that are different than other populations of cells in that lump. By the time a tumor is 6 millimeters in diameter, the size of a marble, that it can have 1 billion cells, or even many more than that, depending on which reference, which resource you look at, and that those cells, in the center of the cells, there's a decreasing amount of oxygen. It's hypooxygenated or hypoxic. And that, because the cancer cells can adapt, because they can compensate, they now start producing molecules to induce the growth of their own blood vessel vasculature to survive. And that is another hallmark. And in addition to that, the cancer cells are producing different molecules that they would that healthy cells don't produce, that actually induce the migration of some types of immune system cells. You can think of it as hijacking these immune system cells and cause them to migrate and right adjacent to the cancer cells. And those immune cells then provide a type of protection, a type of camouflage, so that the cancer lump, the tumor cells and the cancer stem cells are able to avoid immune destruction. And so let's look at the microenvironment of the tumor, where it's not just cancer cells. Here's a tumor microenvironment. So in this graphic, the pink cells are the cancer cells, and these other cells that are illustrated, here are the blood vessels that have been induced to grow by the cancer cells. And these other cells, some supporting cells, some immune system cells, which you see here, this MDSC cell, this T regulatory cell, this macrophage, it's actually a type 2, it's polarized to a type M2 macrophage. You can think of them as bad guy pro-cancer immune cells. So it's helpful to look at the immune system to understand how this can occur and see the immune system as having 3 categories of function. The first category, immune surveillance, danger monitoring, good danger monitoring, and that's represented in this graphic by this cell right here. Think of these immune danger monitoring cells as sentinels, as alarm cells, and they're widely distributed, billions of them throughout your body in every compartment of the body, and they're constantly rolling along. Think of them as molecularly sniffing for danger, which is trauma, poison, infection, and cancer, just broadly. And that when they encounter danger, they immediately activate an alarm response. And that alarm response is to activate and notify the good danger containment system, which is the next category within the immune system for this understanding. That the good danger containment, you think of it as good inflammation, which includes good anti-cancer inflammation, has several types, cell types identified in this graphic. And this, by the way, is an old graphic. It's over 10 years old, and I still use it because I think it's very helpful for understanding and gets the overview of these 3 categories that are relevant. And in the good danger containment system, there are subcategories: the immediate responders in the innate immune cells, immune system, and they're already pre-positioned as well, that they're ready to respond to danger when activated immediately. And that then there's also the delayed responders, the adaptive immune cells, and these include T cells and B cells that can be back in the lymph nodes that are then stimulated to multiply, and they can multiply in the billions, millions and billions, enough to respond to contain whatever the necessary, or to contain whatever danger and the amount necessary to contain that danger. And then when that danger is contained, we need to turn off the good, desirable inflammation, and that's where the next category, the good anti-inflammatory wound repair and clean up and restore response. And there's 2 of the numerous cell types are identified right here, and that these essentially counteract or balance the good inflammation with good anti-inflammation. And when you think about wound repair implies that you're going to regrow, restore the tissue. And so wound repair is producing growth signals, growth molecules to regrow cells, and the cancer cells exploit this. So this category, these 3 categories help understand how the exploitation of the immune system occurs from the cancer cells. The cancer cells are producing molecules. Sometimes the molecules that they produce are then put out onto the cell membrane of the cancer cell, which identify the cancer cell as malignant. And the good anti-cancer immune cells can see them, identify them, and eliminate those cancer cells. And that's a good thing and desirable. At other times, the cancer cells are producing soluble immune messenger molecules, so soluble chemokines that are released out into the bloodstream and in the serum that literally induce the migration. Think of it as kidnapping these good anti-inflammatory and wound repair cells and forces them to travel up the concentration gradient. It's called chemotaxis, to take up a location immediately adjacent to the cancer cells, where they produce their normal function, where they are producing good anti-inflammatory molecules, which are now bad anti-inflammatory molecules because they suppress the good, desirable anti-cancer immune cells. So think of it this way, that these molecules produced by the cancer cells capture and induce the function of these good immune cells that are now bad guy pro-cancer immune cells, so that when the good anti-cancer immune cells come floating along looking to identify the cancer cells and eliminate them, these guys suppress these guys so that then the cancers remain camouflaged and thus avoiding immune destruction. So in the tumor microenvironment, there are these bad guy pro-cancer immune cells and other immune cells that are in the wound repair, that provide the wound repair function, that are producing molecules to grow like they would in repairing damaged or tissue that needs to be regrown. Well, now the cancer cells are exploiting those growing signals. And that's a big, important understanding about the tumor microenvironment. And that some of those cells are these that are identified here, the MDSC, the myeloid-derived suppressor cells, the T regulatory cells, and the M2 macrophages. Macrophages can polarize to be M1 anti-cancer macrophages or M2 pro-cancer macrophages. So it's important to understand that we're always dealing with and have to consider this tumor microenvironment. So by the time this tumor is large enough to be seen in any kind of imagery, in any kind of clinical evaluation by MRI, CT, or otherwise, that it by definition exists as a microenvironment, and that the normal healthy balance of pro-inflammatory and anti-inflammatory immune messenger molecules in the tumor microenvironment is altered in an adverse way, in a pro-cancer way. And then the final hallmark, metastasis and tissue invasion. And so that because of this inability to stop growing, this lack of sensitivity to turning off the growing signals, when the cancer community, that tumor leaves one organ and hits an adjacent organ, it just keeps going. And anywhere along the way in this sequence, the cells can lose their attachment to the tumor population and release and roll off and float and go to another location, which is the well-known metastasis. And so now looking at that, at these common characteristics, gives us a little more context to understanding there are therapeutic agents that are already available, targeted biologics and some chemotherapies that can target each one of these hallmarks, but they don't target the broad spectrum of the hallmarks. And there are natural products that can target each one of these molecular, each one of these hallmarks. But importantly, there are no natural products that in isolation or in just a small combination can effectively target the range of hallmarks. The same is true with repurposed pharmaceuticals. I'm talking about primary care pharmaceuticals, not the repurposing of conventional oncology drugs or targeted biologics, which can also target the hallmarks. Here I'm talking about non-oncology drugs that are primary care drugs that can also target some of the, or can target the hallmarks, but none of them do so effectively in isolation or in small combinations. So how do we target this, the hallmarks of cancer? Well, it's first by understanding that we need to target molecular pathways in molecular networks concurrently in the cancer cells and in the tumor microenvironment cells, which is what the hallmarks are describing or expressing. And that by looking at these in big patterns of traffic, molecular signaling traffic, comparing healthy traffic in healthy cells to disease traffic in the cancer cells, gives us a way to look into this information. And to understand molecular activities in molecular maps is useful to use skills we already have in looking at regular maps, street maps, Google Maps, the maps that we use every day. So when you're looking at molecular maps, think of each molecular site or molecular target as a molecular address, and it'll be helpful in navigating in these maps. And if I show you a regular map from a high view down to a lower view, down to a even lower view, as you zoom down into this, you get greater and greater detail in that map, and that you're not disoriented. You understand that the more you go into it, you can get greater detail, or that you could come out and have regional views and look at regional information. The same is true in the molecular maps. Here is the most highly referenced, highly cited maps called the Kyoto Encyclopedia of Genes and Genomes, the KEG map, Pathways in Cancer. And notice within this map, and this is a high view, this is a high regional view, that it has a number of the hallmarks identified. Here's sustained angiogenesis, the blood supply. Here's avoiding automatic programmed cell death. Here's that proliferation, division, accumulation, growing division, accumulation, growing division, accumulation. Here's the DNA mutation, the DNA instability, the lack of sensitivity to turning off the growth signaling, the resistance to chemotherapy, the tissue invasion and metastasis. And it gives you an overview without having to know all the details that would be if you zoomed in. So it's akin to this view on a map where you're seeing the regions in this particular map. The next is, let's look at the highways. So if I look on this map and now just focus on the avenues of traffic, here on the KEG cancer map, the avenues of traffic are these molecular pathways that are labeled with terms that may be unfamiliar: hedgehog signaling pathway, notch signaling pathway, VEGF signaling pathway. That the labels for these pathways are important just to recognize as labels. We don't have to know how they came about any more than we know how the highway names, why this one is Highway 30 or this one is Highway 20 or this one's 820. Doesn't matter. We know we can use the map to get to that pathway, that traffic avenue. Same thing here. We can use the labels to go through these pathways. And that if we look at here, again, those highways and zoom in on the various features within, here's sustained angiogenesis. Let's just look at that particular combination. Sustained angiogenesis, and the pathway most highly associated with it, called the VEGF pathway. But notice there's more than just one pathway contributing to angiogenesis, and that by definition means it's a molecular network activity that's expressed in that particular feature, that particular hallmark. And that's the take-home message. Every hallmark is the result of molecular network activity. So let's explore that a little further. When we zoom out on this big map and look at these hallmarks, how are we going to target them? Where do we go? What's the priority? How do we address those in an appropriate way? Well, importantly, that got addressed about 3.5 years ago when the Pan-Cancer Atlas was published. The Pan-Cancer Atlas, after about 10 years and hundreds of researchers looking at many, many thousands of cancer specimens, came out with a prioritization called the Pan-Cancer Atlas flagship paper pathways, signaling pathways. They identified 10 pathways that have particular relevance in every molecular target, every molecular site, every molecular address within those pathways. They're identifying as relevant, as potentially actionable alterations, targetable alterations, in order to give researchers and clinicians the opportunity to target and to combine therapies. And so it's useful to go from this map to a prioritized map, essentially a prioritized subset. And that's what this is, the Pan-Cancer Atlas flagship paper pathways. And if we look further and populate these pathways, we see that there's a lot of molecular addresses that get complicated very quickly. So to make it more accessible, looking at it in an animation provides context. Look at this particular pathway, and that this pathway is associated with proliferation, a hallmark, and cell survival, and the production of protein. And that this sequence, this molecular cascading sequence, is what's associated with these characteristics. This pathway is associated with the ability to respond to adverse oxidative stress. This particular pathway is associated with cell growth, and that there's also a cascade of molecular signaling. And if I fast forward through all of them, you can see that each one of these has particular features associated with it, characteristics associated with it. And then the most important feature of all of them is they're connected. They're all connected. And that has profound implications, because we can then, when looking at molecular targeting, and you're targeting one or another address, molecular site, and then the cells attempt to compensate, we can look at targeting other molecular sites downstream or sideways, the ability of the cancer cell to compensate through compensatory signaling, through adaptation. We can also look at how to target some of those additional molecular sites. So targeting molecular networks and combinations. So then let's look forward at 4 natural products, natural product supplements. Curcumin, the most widely researched natural product in the cancer literature. And notice here in this network, this pathway, remember, this is not a random network. This is the Pan-Cancer Atlas flagship paper pathways network. And that curcumin with the yellow dots throughout the network, it is a prime example of a single therapeutic agent having multi-targeted anti-cancer activity. So it's a single agent that is molecular network targeted. And that's a profound implication. And it's one of the great values of natural products. One of the great limitations of natural products is that they are so poorly bioavailable. But by the time you can take enough of a single natural product therapeutic agent that's multi-targeted, that you can't take enough and tolerate it to have therapeutic effect. By the time you take enough to have a therapeutic effect, it's now not well tolerated. You're either nauseous, sick to your stomach, getting loose bowels, some other adverse effect. So there has to be another way to obtain the benefit from these natural products. And so look here, here's green tea extract, EGCG. Green tea extract, also another multi-targeted therapeutic agent, another molecular network targeted agent. And if we look right here at this molecular address, green tea, curcumin, green tea, curcumin. Green tea, curcumin. They overlap. Both those two different therapeutic agents affect those molecular sites in the same anti-cancer direction. And that's of great value because in the literature, when it shows that there's overlapping therapeutic effect, that synergistic effect, that the amount of each of the two items necessary together can be dramatically lower than the amount separate and the amount in isolation. It can be half as much needed. It can be even 1/10 as much needed. It can even be 1/100th amount needed. So an advantage of 1 to 2 orders of magnitude, which are then very practical, therapeutically available doses that people can tolerate day in and day out for extended duration. And now let's look downstream of these molecular sites. And here, curcumin green tea, curcumin green tea. And at that point, they're not the same. They're not targeting the same molecular sites. They're now targeting different. So we're spreading out the molecular targeting. So by combining these two, we're engaging molecular synergy at some molecular sites, at some molecular targets, and also expanding our molecular network coverage. And that's of great value. Now let's look at 2 more items. Genistein from soybeans. Genistein is so well known as an anti-cancer agent. It is a reference for anti-cancer research. Notice the pink dots throughout the molecular network. So genistein is also a molecular network targeted anti-cancer agent. And then resveratrol. Resveratrol from grapes, from peanuts, from pine trees, from a number of other plants. It has a very broad and robust literature for molecular network targeting of anti-cancer intervention. Remember, flagship paper pathways, not just any pathways, but each one of these molecular sites is a potentially actionable alteration. And then combining these items, and there's the real value and the breakthrough for molecular integrative oncology, is everywhere you see multicolored dots at a molecular site is an opportunity for therapeutic overlapping anti-cancer synergistic activity. And that's where we're aiming through this to control molecular signaling networks with molecular network targeting using these evidence-based overlapping therapeutic agents. Here's that same data in a more conventional table. The first column is looking at every one of these flagship paper molecular targets, and that every row where you see more than one check mark is an opportunity for overlapping therapeutic anti-cancer synergy. So let's look at some additional molecular maps that demonstrate just visually about all of the hallmarks as being molecular networks. And importantly, this is not something to memorize because you can always go and get the information through the molecular maps any more than you would memorize a map on Google Maps, knowing that you can always go to the Google Maps to get this information. I'm demonstrating this information to show that each one of the hallmarks is molecular network generated, and then to take an example of just one or a small group of molecular sites that are in the flagship Pan-Cancer Atlas Pathways network and show how we can zoom in on those within the various hallmarks. And so here's the first hallmark, genome instability and mutation, lots of molecular activities. And if we zoom in just on one molecular site or complex, we could see, and this is in the Pan-Cancer Atlas Pathway. Same thing for pro-cancer metabolism or deregulating cellular energetics, zooming in on a particular well-known molecular site. Just because I'm seeing it in this pathway in this hallmark doesn't exclude it from other hallmarks. Remember, the pathways are all interlinked, they're all overlapping. Molecular networks are integrated among each other. the sustained proliferative signaling, the ongoing pro-growth signaling. We look at mTOR is part of that network. Evading growth suppressors, another molecular network. We zoom in on a complex called SMAD2, SMAD3, SMAD4. These are just examples. They're not, I'm not identifying any particular hierarchy among these molecular sites other than that they're in the pan-cancer atlas pathways. Resisting cell death and an important tumor suppressor, P10. Enabling replicative immortality, another molecular network. And we're looking at a complex of Notch 1, 2, 3, and 4. It's also part of the pan-cancer atlas signaling pathways. Inducing blood supply. In addition to VEGF signaling, which is well recognized and described right here, also other HRAS, RAF1, and MAP2K or MEK. Pro-cancer inflammation, well recognized in multiple pathways, in multiple hallmarks, PI3K, AKT. Avoiding immune destruction. Raptor mTOR complex, well recognized and in other hallmarks as well. Activating invasion and metastasis, another molecular network. CTNNB1. In the point is, think molecular networks. That's what matters. That's what's relevant. And that's what we need to target. In order to control the progression of the cancer, we need to control molecular networks, the molecular signaling in cancer cells and tumor microenvironment cells in a manner that's evidence-based and well tolerated for extended duration. In contrast and in parallel to the conventional chemotherapy or biologic targeted agents, which have a superb capability of eliminating cancer cells and controlling molecular activities, but generally only once in a while, especially with the cytotoxic chemotherapy, but that have a potentially higher adverse risk profile. So integrative oncology, this organization, the Society of Integrative Oncology, made the definition. This is a landmark definition that defines it for the field. Is a patient-centered, evidence-informed field of cancer care that utilizes mind and body practices, natural products, and/or lifestyle modifications from different traditions alongside conventional cancer treatments. What I'm focusing on is the natural products alongside conventional cancer treatment. And precision medicine, defined by cancer.gov and NIH, essentially says that using information about a person's genes, proteins, environment, and lifestyle to prevent, diagnose, and treat disease in the individual and the variability in the individual. And if we take that information, remembering that precision medicine, looking at the genes and proteins, DNA is transcribed into RNA, is translated into protein, those big three, genomics, transcriptomics, and proteomics, and we add the foundation of integrative oncology, that's how we get molecular integrative oncology, targeting the molecular networks that are relevant in an individual's particular cancer type. And combining these definitions as we've seen them defined by the Society for Integrative Oncology as well as cancer.gov and NIH to get this definition. Molecular Integrative Oncology is a patient-centered, evidence-based field of cancer care that utilizes precision medicine information about a person's genes expression, proteins, environment, and lifestyle to determine molecularly targeted anti-cancer natural product treatment recommendations, in addition to, not instead of, conventional cancer treatments, including chemotherapy, targeted biologics, and radiation therapy. And importantly, natural products have some significant advantages and benefits, as well as significant disadvantages and limitations. The advantages is that they inherently target molecular networks. That's of vital, fundamental importance, because the cancer hallmarks, the cancer activities, are inherently the activities of molecular networks. So we're using molecular networks to target molecular networks. They demonstrate in vivo and in vitro synergistic anti-cancer activity and provide a rational therapeutic extended duration control of cancer molecular networks with a relative risk and margin of safety that's acceptable and manageable. And they're immediately available on the market for patients. The problem, or one of the great, or these great disadvantages, is that they are very poorly bioavailable and very limited in their absorbability and their absorption, and that the products are often inconsistent. There's inadequate product consistency and dosage forms and control of those for the consumer, and that inherently they have a low therapeutic impact when utilized separately or in limited combinations with very limited clinical trial data in humans, and virtually nil funding for clinical trials, especially of this broad spectrum of anti-cancer combinations for controlling the molecular networks, and that the peer-reviewed data is most often limited only to in vivo and in vitro anti-cancer findings, basically human and animal cancer cells in animals and test tubes, preclinical data rather than clinical data in humans. And so we need to take both the advantages and the disadvantages into account when making any kinds of clinical recommendations for patients. That's the practical understanding of the hallmarks of cancer and molecular integrative oncology. Hopefully you then have the ability to now look at these with much greater understanding, a solid foundation, and be able to explore more deeply as it is appropriate for you and with your patients or with your family members or in discussion with your physicians. So I'll now take any questions, and also encourage you that if you don't get your question asked, or if you'd like to reach out directly, please do so. I'm happy to respond offline, so please feel encouraged to reach out at any time. Thank you for your attention. I look forward to whatever your questions.
Thank you, Dr. LaValle. Reminder for everyone, if they have any questions, to please post in the Q&A box of your Zoom menu. We did receive a question asking if the webinar will be recorded, and it will be. But we're going to have it up on the SIO YouTube channel, hopefully by tomorrow. So a post will be out on social media, so please be sure you're following us on media to be able to find the link to the recording from today's webinar. Let's see, we'll wait a few minutes. People are typing.
This is the presentation that I wish someone would have been able to give to me when I started out. It would have made it a lot easier to understand because the sequence of events puts some logic to understand step by step what's happening in the hallmarks of cancer.
Okay, here they are flowing in now. Okay, first one, can MIO help make conventional treatments more effective?
Good question. So there aren't clinical trials that show that. My clinical experience, however, shows that because the data demonstrates that depending on the chemotherapy, the cytotoxic agent, there are data supporting the increase of the anti-cancer benefit from that chemotherapy. So it increases the sensitivity of the cancer cell to be eliminated or controlled by the chemotherapy, and concurrently avoid interfering with the molecular mechanism of action of that chemotherapy. And remember, and I didn't emphasize this, that we're talking about targeting the cancer. So there's also direct anti-cancer activity from these natural products. And there are also some not cancer activity benefits in healthy organs and systems. So natural products have off-target, if we think of the cancer as the target, that they have off-target benefits in other organs. So cardiovascular, respiratory, the gastrointestinal, central nervous system, immune system can have benefit from some of them. So the net result is appropriately selected evidence-based natural products and repurposed pharmaceuticals based on the particular, whether it's chemotherapy or targeted agent, can have significant additional benefit. That's what we're attempting to test within clinical trials in the organization called broadspec.org, but unfortunately we've gotten no funding yet to do so. If anybody has any encouragement or possibility of funding, please contact me for that.
Great, thank you. Next one is how to deal with the low absorption issues.
I'm sorry, say that again.
How to deal with the low absorption issues.
Yeah, that's a major problem, major challenge. So the sometimes having them modified so that you increase the solubility. For instance, curcumin is soluble in oil. It also dissolves in boiling water if you boil it for 10 to 12 minutes. Other items, there are some other natural products that can be combined with them that increase the bioavailability. The most important feature, however, is the evidence-based overlapping selection. So making appropriate selections so that there are natural products that have synergistic effects, and that those synergistic effects can increase the value of the therapeutic impact by even up to 1 to 2 orders of magnitude. So that's really where the greatest value comes in, is looking for overlapping synergies.
Great. And last question that we have, repurposed meds / non-oncology, do you see these working synergistically too? And then another question, are there ones you would go to?
Yeah, there are data showing that the natural products and let's be clear, we're talking about in each case specific selected natural products or specific selected repurposed pharmaceuticals that have in vivo and in vitro demonstrated evidence for synergy with various types of chemotherapy, cytotoxic agents, sometimes with the new targeted biologics, but not often because they're so new on the market and there's not been a lot of research looking at them, as well as the repurposed pharmaceuticals, some of them having synergy with chemotherapy agents, as well as the natural products having synergy with radiation therapy, so enhancing the sensitivity of the cancer cells to be killed by radiation, and concurrently reduce some, not all, some of the adverse effects, some of the adverse harm to the healthy cells and organs. It's called cytoprotection, CYTO, cytoprotection. And there's a broad range of data looking at the repurposed pharmaceuticals, and you could see the repurposed pharmaceuticals. Sometimes there are data showing synergy between the natural products and the repurposed pharmaceuticals as well.
Fantastic. Okay, we have a bunch of thank yous in the chat to Dr. LaValle, and I think that's all.
Attention and the opportunity. I hope it's helpful. Please reach out if there's any questions or you'd like to discuss any of this at any time.
Yes, I did put your website in the chat. Your website in the chat for anybody to be directed to. And let me just see. Yep, everybody's just saying thank you. Okay, fantastic. Well, thank you, Dr. Valley. It was a pleasure having you with us today.
Bye. Thank you very much.